So I mentioned in the photosynthesis video that cellular respiration, which occurs in the mitochondria, is kind of the reverse process of photosynthesis. Chemically, it's the reverse reaction, and respiration is taking in oxygen, letting out carbon dioxide. Well, respiration doesn't just occur in the mitochondria, it occurs in chloroplasts too. The respiration that takes place in the chloroplast is called photorespiration. Shouldn't be too hard to remember. Photosynthesis, photorespiration, they both take place in the chloroplast. So, let's have a look at the chloroplast again. Okay, this is where photorespiration occurs, in the stroma of the chloroplast. Same as the Calvin cycle, because it's actually when oxygen interrupts the Calvin cycle by binding with rubisco instead of allowing carbon dioxide to. And actually this C and O in rubisco stands for carboxylase oxygenase, meaning that rubisco can bind with either carbon dioxide or oxygen, and it's the relative concentration of each that determines which gas rubisco binds to. Normally in the Calvin cycle, when rubisco binds with carbon dioxide, the first product is a 3-carbon compound called phosphoglycerate, or PGA. And 2 of these get made per cycle, which go on to become 2 G3Ps, the building blocks for glucose. This regular Calvin cycle is called the C3 pathway because phosphoglycerate, the first product of the cycle, is a 3-carbon compound. What happens when rubisco binds with oxygen instead of carbon dioxide is that we get only 1 phosphoglycerate molecule instead of 2, and 1 phosphoglycolate molecule instead. And phosphoglycolate doesn't go on to manufacture glucose like phosphoglycerate does. In fact, phosphoglycolate eventually gets converted back into carbon dioxide in the mitochondria. So this is undoing the work done so far to fix this carbon, and it's reversing this Calvin cycle, and it's using energy to do this, so it's considered a wasteful process. Now, some plants have adapted another pathway to avoid this photorespiration problem. Photorespiration is when rubisco binds with oxygen instead of carbon dioxide. And this alternate pathway that some plants have evolved is called the C4 cycle. You might have guessed by now that if the C3 pathway has a 3-carbon compound as its first product, then in the C4 pathway the first product must be a 4-carbon compound, and you would be right. Oxaloacetate is the 4-carbon compound in the C4 pathway. And malate gets made instead of glyceraldehyde 3-phosphate. This is an important component of the C4 pathway, so remember this compound here, malate. It's kind of key to the whole C4 cycle. And this malate-producing reaction is catalyzed by, instead of rubisco, it's catalyzed by PEP carboxylase, which fixes carbon dioxide like rubisco, but unlike rubisco, PEP carboxylase binds carbon dioxide to PEP. And PEP stands for phosphenolpyruvate. You can call it PEP. PEP carboxylase cannot bind with oxygen. It only binds with carbon dioxide. And this is how C4 plants avoid photorespiration. PEP carboxylase is the defense against photorespiration. However, if we've still got rubisco hanging around, then rubisco can still bind with the available oxygen and continue to undergo photorespiration. So just having PEP carboxylase is not enough to prevent photorespiration. We also have to isolate rubisco from oxygen, wall it off somehow, and C4 plants have found a clever way to do just this. Their solution is in their leaf structure. So let me show you how they're different from C3 leaves structurally. But first let me add that unlike in the C3 pathway, which occurs entirely in this one organelle, the C4 pathway, this cycle here, takes place between two adjacent cells. These are the two adjacent cells where the C4 pathway takes place, in the green mesophyll and red bundle sheath. You're looking at a cross-section of a C4 leaf and C3 leaf. Notice all this air space containing carbon dioxide and oxygen, and how in the C3 leaf, bundle sheath cells are exposed to this air. And rubisco is found in all of these cells, the mesophyll and the bundle sheath. Mesophyll cells are marked with an M and bundle sheath with a B. I made the C4 bundle sheath cells red to highlight that rubisco is isolated here, whereas in C3 plants, rubisco is everywhere, exposed to this airspace where there's oxygen. Bundle sheath cells are cells that surround the veins of the plant, which I drew in blue. Notice that the bundle sheath cells themselves are surrounded by the mesophyll cells in C4 plants, and this in effect walls off the bundle sheath from the oxygen in this airspace. Here, the only gas that rubisco has access to is carbon dioxide, so there's no photorespiration. Now, if rubisco is buried deep in the C4 plant and shielded from the airspace, how does carbon dioxide get to the shielded rubisco? Well, remember I mentioned that an important product of the C4 cycle is malate. Malate is like a carbon dioxide carrier or transporter. You can think of it that way because in the mesophyll, PEP carboxylase traps carbon dioxide in malate, and this malate then gets shuttled from the mesophyll to the bundle sheath cells where carbon dioxide is finally released for rubisco to process in the Calvin cycle here. While PEP carboxylase is exposed to this airspace being in mesophyll, PEP carboxylase has no affinity for oxygen. It will only bind with carbon dioxide. Therefore, only carbon dioxide gets fixed and delivered to rubisco in the bundle sheath cells. In C3 plants, rubisco is everywhere, but in C4 plants we have PEP carboxylase fixing carbon dioxide here and rubisco making sugar there. Two different enzymes doing two separate things in two separate cells. We can't get rid of rubisco to avoid photorespiration because we still need rubisco for sugar production. We just have to keep it away from oxygen for it to be more efficient. And C4 plants do this by having rubisco contained only in the cells that are not exposed to this airflow, where there's both carbon dioxide and oxygen. These specialized cells in the C4 pathway separate oxygen from rubisco by using PEP carboxylase in the initial carbon fixation step instead of rubisco, so that rubisco only makes phosphoglycerate and not phosphoglycolate, the wasteful product. In the C4 pathway, we just have this additional step of trapping carbon dioxide and malate in the mesophyll before the Calvin cycle can resume in the bundle sheath. By the way, the C3 pathway is the Calvin cycle, not to confuse you. It's in hot, sunny climates where we most find C4 plants, and here's the reason. Plants have pores called stomata that allow for gas exchange, but unfortunately, the stomata also allow for water to be lost. In high temperatures, plants will close their stomata to conserve water, but this creates a buildup of oxygen. Because remember, oxygen is a byproduct of photosynthesis, and as oxygen levels increase inside the plant, more photorespiration will occur because more oxygen will bind with rubisco. So you can imagine that this C4 adaptation was in response to intense light conditions, high heat and little water, plants having to close off their stomata and not letting out the oxygen. Most plants are C3 plants. C3 plants are more frost resistant and less geographically restricted than C4 plants. However, when it comes to high temperatures and strong light intensities, C4 plants often outcompete C3 plants because photorespiration is really counterproductive in the hot, sunny climates where C4 plants are found. In these regions, C4 plants close their stomata to conserve water, which builds up the oxygen. Therefore, high light intensities favor C4 plants. Some examples of C4 plants are corn, sorghum, sugarcane, millet, amaranth, Bermuda grass, crabgrass, most tropical grasses, but some shrubs too. Now the third kind of plant, CAM plants, also utilize the C4 pathway, and you could consider them a type of C4, but instead of spatially or physically separating oxygen from rubisco, CAM plants reserve carbon dioxide in their vacuoles, in their storage vesicles. And they do this at night so that they have it to replenish in the cell when carbon dioxide levels drop during the day, because it's when carbon dioxide levels drop low enough that photorespiration occurs more than photosynthesis. At a very low level of carbon dioxide, oxygen will outcompete carbon dioxide for rubisco, and the plant would be using more fuel than it's making. So the strategy of CAM plants is not to isolate rubisco from the space where gas exchange occurs, but CAM plants instead just keep carbon dioxide levels high to avoid photorespiration. Unlike C3 and C4 plants, CAM plants close their stomata during the day and open them at night to take in carbon dioxide. But it's during the day when they need this carbon dioxide, so that's why CAM plants have to store the carbon dioxide they collect at night, and they store it in their vacuoles in the form of malate or aspartate. You see, CAM plants are typically desert plants, and water loss is greatest during the day. That's why CAM plants close their stomata during the day, which is the opposite of what most other plants do. C3 and C4 plants take in carbon dioxide at the same time as when the light reaction occurs during the day, when there is sunlight available to power photosynthesis. Because the dark reaction uses the products of the light reaction, C3 and C4 plants take in carbon dioxide at the same time that the light reaction is occurring. But CAM plants simply can't afford to do this. They can't afford the water loss that would occur if they left their stomata open during the day. They too have to wait for the daylight to start the light reaction, but unlike C3 and C4 plants, they're not getting their carbon dioxide during the day. They're getting it at night, storing it in the vacuoles, and therefore CAM plants typically have larger vacuoles than other plants. While light is the primary consideration in C4 plants, CAM plants respond to temperature, humidity, and salinity, which makes them more drought tolerant. They are most concerned with water loss. CAM plants are pineapples, air plants, some orchids, many desert plants, succulents such as cacti, jade plants, yucca, agaves, and aloes. Keep in mind that all three plants utilize the C3 pathway, the Calvin cycle, which is the pathway required to make sugar from carbon dioxide. The only difference is that C4 and CAM plants have additional mechanisms for sequestering carbon dioxide and reducing photorespiration. Because photorespiration reduces net productivity of the plant, there is interest in altering crops to carry out less photorespiration in order to increase crop yield. Some strategies might include genetic modification of rubisco to increase specificity for carbon dioxide relative to oxygen, or increasing carbon fixation via another enzyme. It's estimated that 15 to 40% of the carbon fixed in photosynthesis is lost through photorespiration, depending on the plant species. So this is an area where some efficiency can be recovered. Interestingly, you can find the CAM adaptation in aquatic plants as well. Not due to the lack of water, obviously, but because carbon dioxide diffuses much slower in water. Levels of it can vary. The CAM pathway allows aquatic plants to store up or sequester carbon dioxide, just like in terrestrial plants. So two very different needs, but one biochemical mechanism solves both. the CAM strategy of reserving carbon dioxide in malate in vacuoles. It should be noted that not all plants are exclusively C3 or C4 or CAM. Some plant species can be induced by environmental changes to switch between C3, C4, and CAM. Certain succulents, for example, can do this. And some plants can operate more than one of these mechanisms at the same time, like C3 in the leaves and CAM in the stems. Plants have evolved many ways to live in the environments they live in. And that's it for this video. I welcome your questions and comments below. Please let me know if this was helpful. Thank you.